Hello everyone, welcome for another chapter of Understanding Biology, Chapter 22, Systematics and Phylogeny. As you learned previously about evidence for evolution in Chapter 20 and 21, now we are going to understand systematics and phylogeny in this chapter. So, when you think about natural selection, some examples of natural selection such as Darwin's finches actually tell us about how organisms are evolving through using natural selection. Most direct evidence for evolution comes from fossil. So based on that, we now know that all species arose from a common ancestor. So therefore, all organisms share many biological characteristics together. Some of these shared biological characters include having one or more cells, store genetic information in nucleic acid, etc. So one of the most important tasks for biologists is to group organisms based on their shared characteristics. So the problem with these tasks is that imperfect fossil records. We cannot find perfect fossil records for all the species that we have today. Because of this, scientists are left to rely on additional type of evidence to establish the best hypothesis for evolution or evolutionary relationships. We call these hypotheses because they can be tested based on evidence that we have, and then when new evidence become available, these actually can be proved wrong and new hypotheses can be developed based on the new evidence, and these also are open to be tested. So when you actually reconstruct evolutionary relationships using evidence, this is called systematics. So we use systematics to analyze similarities and differences of a species, and using that we can actually construct an evolutionary tree or phylogeny. A phylogeny is actually a hypothesis about the patterns of relationship among species. This actually represents evolutionary relationship among the species that you are interested in. Therefore, now you know the difference between systematics and phylogeny. Charles Darwin is the first person that came up with this idea of using branching patterns to depict evolutionary relationships. He envisioned that all the species were descended from a single common ancestor. In a phylogenetic tree, therefore, the twigs represents existing species. As you go down the tree, joining these twigs and branches, it represents pattern of common ancestry leading to a single common ancestor of all life. In this figure to the left, you can see Darwin's own diagram of depicting the history of life as a branching pattern of a tree. When constructing phylogenetic tree, looking for similarities between a species may not always be a good predictor. So this is because when you look at similarities shared by species, it has been once two species is diversified, it's not a very good character just to look at similarities. This is because evolution never occurs in a constant rate. Sometimes evolution occurs in a very rapid manner. Sometimes it can occur in slowly manner. Therefore, as you learned previously, evolutionary is not also unidirectional. And also you already learn about convergent evolution. Convergent evolution can happen where species evolve convergently due to their similar habitats. Even though they are apart by long distance, they may have similar characteristics. One of the examples of convergent evolution is that marsupials and placental mammal. This could lead to two species that are not related to each other to have similar characteristics. So therefore, looking at similarities to develop a phylogenetic tree is not always accurate. So these are the several problems that we talked about: the rate of evolution, and evolution is always not unidirectional, and it's not always divergent. Can convergent evolution happen? So looking at similarities may not be a good indicator for understanding evolutionary relationships. When you develop evolutionary relationship, one of the earlier techniques systematics use is cladistics. So we are going to look at how we can actually use cladistics to understand evolutionary relationships and to develop a cladogram. So before we go into that, let's talk about derived characteristics. So like we discussed earlier, due to the reasons of evolutionary rate, unidirectionality, not being unidirectional, and also not being always divergent, most systematics no longer use phenotypic similarities to construct phylogenetic relationship. Therefore, the best way to reconstruct phylogeny is to distinguish characteristics inherited from most recent common ancestors. These type of shared characters are called derived characters. So derived characters are the ones that we normally use for constructing evolutionary relationships. So if you have derived characteristics, you also have ancestral characteristics. Ancestral characteristics are the similarities that arose prior to the common ancestor of that group. So one of the important requirements of cladistics is to be able to distinguish between derived and ancestral characteristics. So in cladistics, we only use shared derived characters for evolutionary relationships. So the characters that we use in cladistics can be morphological, behavioral, physiological, or molecular, such as DNA, RNA, or protein. And this process requires to identify character status as ancestral or derived. First, it is important to gather data on a number of characteristics for all the species in your analysis. And once you gather all of this, you look one by one of each species, each characteristics, and you are going to recognize them as ancestral or derived, present or absent. So when you look at how to distinguish between ancestral and derived characteristics, one of the example is presence of hair in mammals. So presence of hair in mammals is a derived feature. Presence of lungs in mammals is an ancestral character because it has been there in earlier organisms such as amphibians and reptiles. Therefore, it evolved prior to the last common ancestor of mammals, hence considered as an ancestral character. Shared derived feature of hair suggests that all mammal species share a common ancestor that existed more recently than the common ancestor of mammals, amphibians, and reptiles. Once you assembled your data, the first step of manual cladistics is to polarize your characters. So this means that you need to determine whether a particular character is in ancestral or derived state. If it is ancestral state, you give a 0. If it is in a derived state, you give a number 1. For example, for teeth, you must see if the teeth is present or absent in most recent common ancestor of this group. So after you do that, you give a numbering system such as this shown in this figure, and you do this for all the species in your analysis. So when you are constructing cladogram, it is very important to consider an ancestral group or a fossil record as your ancestral group, because you need to first compare what are the characteristics that are present in these fossil records. But sometimes you all know fossil records are not perfect. You might not be able to find a perfect fossil record for all the characteristics that you are observing for these several species that you are interested in. In that case, you can use this method called outgroup comparison. So outgroup comparison, you need to find a species or a group of species that is closely related to but not a member of the group under the study. And you designate it as the outgroup of this study. For this particular tree that we are looking at, lamprey is used as the outgroup. So once for each character such as present of jaws, lungs, amniotic membrane, hair, no tail, and bipedal, you look at all of these characteristics in the group of outgroup, which is lamprey here. Since outgroup, they don't have any of these characteristics, you give 0, or ancestral characteristics, you give 0. If it is a derived shared character, you give number 1 for all the rest of the species if they have it. If they don't have it, you don't give that number. Once you do all of these, then you can develop a cladogram. So picture to the left is a cladogram. So cladogram is actually a depiction of a hypothesis of evolutionary relationships. Species that share a common ancestor with the shared derived characters are said to belong to a clade. Clades are evolutionary units and refer to a common ancestor and all of its descendants. So when you look at this cladogram that is depicted here, the square represents the ancestor, and all the tips of these branches are descendants from this ancestor. So the time goes from past to recent. This cladogram is actually sort of like a family tree, as you can see here. So like I mentioned, cladogram depicts a hypothesis of evolutionary relationship, and a clade, you need to remember this word, is a species that share a common ancestor as indicated by the possession of shared derived characters, and evolutionary units refer to a common ancestor and all the descendants. In a clade, you have to include all the descendants and their common ancestor. So a derived character shared by a clade member is called a synapomorphy of that clade. Let us see how to read a cladogram. In a cladogram, you can see ancestral lineage is shown in a blue. So where a change occurred leading to a speciation event that give rise to a 2 daughter lineages. So the daughter lineages are here in the black color. Here in the square, you can see the speciation event that occurred to separate these 2 daughter lineages. When you look at a cladogram, each lineage has a unique history and also a shared history. When you look at this particular cladogram, from ancestral lineage, B and C contain shared history, while B and C also have their own unique history. The blue color here is a unique history for C, and green color solid line here represent unique history for B. And solid black color line here is unique history for A, and this dotted line is actually showing unique history of B and C in this cladogram. Each lineage also contain ancestors that are unique to it and ancestors that are shared with other lineages. So when you look at here from the common ancestor, there is one common ancestor here that is common for A, B, and C, because they all derived from this common ancestor. And when you look here, this common ancestor that is situated here is shared by B and C. And here there is another unique common ancestor only for C. How do you recognize a clade is another question. Clade is a group that contain a common ancestor and all of their descendants. When you look at the cladogram, we colored it here. So here is a clade. Clade contain their shared common ancestor and all of their descendants here. And you can see this one is not a clade because they are not, all of them didn't derived from a unique common ancestor, which is here. This one is also included, this part together is not a clade. And this one also not a clade because this one not included. If you include this one, this species, then it is a clade. So clade actually include one common ancestor, one shared common ancestor, and all of its descendants. Sometimes you can actually nest these clades to each other. A clade may include many thousands of species or just a few. So you can call this as a clade, this as a clade, this as a clade. So these can be nested. So when you are reading a cladogram, you have to remember that these cladograms are trees. They are not ladders. So they don't show arrows like this, like from A arrows B, from B arrows C, from C arrows D. That is not how you read a cladogram. You have to remember this as a tree, not as a ladder. You also, one of the mistake a lot of people do is that understanding a cladogram where you're reading left to right, you think it is actually advancement, which is not true, because usually the way you place these tips of these branches is not always the same. You can actually arrange it in a way that you prefer. As long as the common ancestor is shared and that relationship is shown, it's not necessarily showing the advancement when you read from left to right. So, which lineage is right or left is arbitrary. Not because we read from left to right. We tend to put whatever the group we are interested in on right. So, these two cladograms are the same. You can see there is no arbitrary selection from left to right of these species. As long as evolutionary relationships are represented accurately, a cladogram can be arranged the way that you are interested in, whatever the species that you are interested in. Let's further look into cladograms. When group of species share a derived character with all the clade members, this is called a synapomorphy. Synapomorphy is a derived character shared by all the clade members. Clade that share amniotic sac is synapomorphy. It's one of the example. So simple cladogram is nested set of clades and each characterized by its own synapomorphies. So here if you look into this cladogram, this amniotic membrane here, this character is shared by all the species up above here. So this is synapomorphy of this clade. So you remember that synapomorphy is a derived character shared by clade members. So this character, amniotic membrane, is shared by tiger, gorilla, humans, and that is called a synapomorphy of that clade. So you can take another example. So you can see hair, which is here, is shared by tigers, gorillas, and humans. So all of these have a hair as synapomorphy of this clade. All of these have amniotic membrane as a synapomorphy of this clade. So now we are going to look at plesiomorphy. In a cladogram, ancestral states are called plesiomorphies. Shared ancestral states with the clade member are called symplesiomorphy. So earlier we talked about shared derived character. Now we are looking at ancestral states. Shared derived character is called synapomorphy. Ancestral state is called plesiomorphy. And ancestral state shared by clade members are called symplesiomorphy. So these are certain words that you need to remember when you are actually understanding a cladogram. So consider the character state presence of tail, which is exhibited by lampreys, sharks, salamanders, lizards, and tigers. Does this mean that the tigers are more closely related to and shared more recent common ancestor with lizards and sharks other than apes and humans? So you can think about that. So symplesiomorphies reflects character states inherited from a distant ancestor. They do not imply that the species exhibiting that states are close related. Just to make sure that when you are talking about symplesiomorphies, you are just talking about its the presence of a shared ancestral characters. That does not mean that they are actually close related because they have symplesiomorphy. Now we are going to look at another character which is very important when you are actually developing cladograms. So when you develop cladograms to understand evolutionary relationship, these can get misled by this homeoplasy. So homeoplasy are actually some characters evolved independently in several species. When we talk about convergent evolution, if you remember, due to geographical similarities of habitats, even though these species are separated 1,000 miles apart, they actually develop similar characteristics. This kind of evolution is called convergent evolution. These characters can be categorized as shared derived characters, which can be misleading in cladograms, because we are actually in cladograms we only think shared derived characters are informative in phylogenetics. So if you have homeoplasy, where a shared character states that has not been inherited from a common ancestor because it was developed due to a convergent evolution or evolutionary reversal, this could be misleading and it could give errors in evolutionary relationships. So it is very important that you need to rely on principle of parsimony to avoid situations like homeoplasy. So principle of parsimony is actually a way to avoid this kind of situation where your evolutionary relationships can get misled by homeoplasy. So systematics actually rely on principle of parsimony, which prefers the hypothesis that requires the fewest assumption. So parsimony favors the hypothesis that require the fewest assumptions. So let's look at what is parsimony. In a phylogeny, you are actually constructing a phylogenetic tree using a cladogram. So when you develop the cladogram, you are actually going to look at which cladogram requires the fewest evolutionary events, and then you choose that cladogram as the correct, accurate cladogram to show the evolutionary relationships. So here I have an example. So if you look at these trees, cladograms A and B, the placement of frogs as closely related to salamanders requires that tail loss occurs twice as an example of homoplasy. If frogs are closely related to gorillas, then the tail loss only had to evolve once. However, this arrangement will require 2 additional evolutionary changes where frogs would have to lost amniotic membrane and hair. Hair could have evolved independently in tigers and humans and gorillas. Now, as you can see, the cladogram depicted in picture A actually requires 4 changes, where the cladograms that is depicted in B requires 5 changes. Therefore, in a principle of parsimony, the cladogram that actually requires the fewest number of evolutionary changes is cladogram A. So that is favored to avoid this problem with homoplasy. So example of cladograms we used so far actually looked at all the morphological characters. With increasing availability of DNA sequencing data, we can now use DNA sequencing data to construct phylogeny. DNA sequence data allow to analyze large number of characters, hence it is more favored nowadays to look at DNA sequencing data for cladogram constructions. So character status are rooted by referring to the sequence of an outgroup, which is distantly related to group to the outside clade, and cladogram is constructed that minimize the amount of character evolution required according to the principle of parsimony. So here is example of DNA sequencing data. As you can see here, we are looking at several 4 species of trees and outgroup. So you acquire all the DNA sequencing data for these species. You can select a specific gene that you are looking for and acquire all of these sequencing data, and they are organized just like the other morphological data, and most parsimonious interpretation of the DNA sequencing data actually used to develop the cladogram. So here in this table, you can see 10 different sites of DNA sequencing data is considered, and outgroup, which is considered as the ancestor, has the DNA sequence data in the very here, and you are actually comparing what are the nucleotide changes in each of these sites in 4 different species that we are interested in. For an example, if you look at this one, the outgroup has adenine as the site A. So species D also have adenine, which is ancestral. Species B also have adenine, which is also ancestral. Now if you look at species C and species A, they have shared derived characteristics, which is guanine in site A. This is how you read the DNA sequences and pluralize them. So now you look at site 2, where the outgroup has a T, which is the ancestral character, and all the rest of them have a shared derived character, which is C. And site 3 have A as the ancestral character, and all of the rest of the 4 species also have ancestral character. This is how you read it. Once you read it, you can actually develop a cladogram based on these nucleotide changes in different sites. So in this cladogram, you look into the site 2, where from the ancestor, the T has changed to a C, and all the 4 species has that change. So therefore, the first branch, the ancestor, has the T converted into a C, which gave rise to all of these species contain that change. So it's a shared common ancestor here that gave rise to all the species here. So once you consider that, the next one is you are actually looking at 2 of the changes here. So the changes that are in green are homologous evolutionary changes. You have to note that. And the changes that are in light blue is homoplastic evolutionary changes. So we talked about homoplasy and why it is actually not good to have homoplasy. That's why we actually have parsimony to kind of like avoid the problem with homoplasy. But if you look in here, this is how you develop the cladogram based on DNA sequencing data. So once you develop the cladogram, you are actually going to look at which cladogram actually requires the least number of changes, and that cladogram is favored for your actual tree. So now that you have an idea of what is the most parsimonious tree and how to develop a cladogram using morphological data as well as DNA sequencing data, now it is also important to understand that sometimes the principle of parsimony may not be always accurate. It could be misleading. Parsimony principle is only accurate if the rate of character change is at a slower rate than the speciation event. But now we know that we found that some characters evolve very rapidly. This is true for genes as well. Certain genes evolve very rapidly, certain genes evolve very slowly. So the rate, evolution rate, can be very different from each other. So we also know different parts of a gene can evolve at different rates. Because of evolution can happen very rapidly, statistical approaches are necessary to infer phylogenetics because parsimony may not be accurate in that case. Therefore, we can actually use different other kind of evolutionary methods that rely on statistics to understand phylogenetic relationships. These are like most likelihood trees that actually utilize statistical analysis to understand the evolutionary relationship. But now we are going to look at how actually we can time the speciation events. So we know that in general, cladograms will only provide the order of evolutionary branching events. They don't inform the timing of the events. So therefore, in order for you to have actually timed event, you can actually use a molecular clock. So evolution actually results from change in DNA. We all know that there are many different variations in DNA called mutations. And mutations, they can be neutral, beneficial, or detrimental. Considering evolution of a molecule is constant through the time, a molecular clock can be developed. And the molecular clock can tell us about the timing of the branching events. So in order for us to use the molecular clock, we need to calibrate it by estimating the time of one of the branching events using fossil or geological records such as formation of a rock, mountain, or a river. So we actually consider that DNA protein sequence evolve at a rate that is relatively constant over the time, and among different organisms we can use this principle to guide us to give a timing for these branching patterns using a molecular clock. So now that we are done talking about cladistics and developing a phylogenetic tree, the next thing we are going to talk about is systematics. So what is systematics and classification. So systematics is the reconstruction and study of evolutionary relationships, and classification is how we place a species in higher groups into a taxonomic hierarchy. Current classification sometimes does not reflect evolutionary relationships. So it is very important to understand how we place species into groups and how we actually should place them into groups based on their phylogenetic relationship. Here are some important concepts to remember. So one is monophyletic group. So what is a monophyletic group? Monophyletic group actually include most recent common ancestor and all of its descendants in a clade is called a monophyletic group. So we also have a paraphyletic group. Paraphyletic group include the most recent common ancestor but not all of its descendants. Here is a monophyletic group. As you can see in this phylogenetic tree, the monophyletic group include the most recent common ancestor and all of its descendant. In this Archaeosaurus clade here is a monophyletic clade. In a paraphyletic clade, you can see this paraphyletic clades has not included all of its descendants, which is excluded here hawk. This is called a paraphyletic group. It does include the most recent common ancestor but not all of its descendants. So we also have a polyphyletic group. In a polyphyletic group, it does not include the most recent common ancestor of all the members of the group. So it does not include the most recent common ancestor. So here is a polyphyletic group, which is bat and hawk together. You can see a polyphyletic group. So taxonomic hierarchies are based on shared traits and they should reflect evolution relationship. So however, traditional taxonomic groups will not fit well with new understanding of phylogenetic relationships, such as birds and reptiles. You know that they are closely related, but if you look at their morphological characters, someone might have hard time understanding that birds and reptiles are closely related because of their morphological characters. So it is therefore very difficult to incorporate new knowledge that we have based on the molecular data and systematics to incorporate into classification. So now that we're done talking about the different kinds of monophyletic, polyphyletic and paraphyletic groups, let's first look at another important factor, which is the species concepts. So in chapter 21, you learned about origin of species. Now you know that biological species concept defines species as groups of interbreeding populations that are reproductively active. In recent years, a new phylogenetic perspective has emerged where now instead of biological species concept, we can actually talk about phylogenetic species concept. This phylogenetic species concept is where the species should be applied to a group of population that have been evolutionarily evolving independently of other groups of population. Phylogenetic analysis should be a way to identify species. In this view, a species is a population or set of populations characterized by one or more shared derived characters. So this phylogenetic species approach actually solves two of the problems with biological species concept. So the first one is the biological species concept cannot apply to all the allopatric populations. because it is difficult to determine whether individuals of a population would interbreed and produce fertile offsprings if they ever came together. But this problem is solved by phylogenetic species concept, because phylogenetic species concept, instead of try to predict what will happen in the future if allopatric populations came together, it look into the past to determine whether a population has evolved independently. So that problem is solved with the phylogenetic species concept. And another problem with biological species concept, it only can be applied for sexual species. But phylogenetic species concept can be applied to both sexual and asexual species. Phylogenetic species concept also have some drawbacks. The first one is it might lead to the recognition of every slightly differentiated population as a distinct species. So this is one of the problems. And another problem is that species may not always monophyletic. Suppose that a population C in the figure here, phylogenetic tree here, somehow isolated and evolved differences that may qualify it to be a species. Because of this, the remaining populations become paraphyletic, even though they might still be perfectly capable of exchanging genes and being a species together. Because of that one species C differentiated, it qualified to be a separate species, made the other four species become paraphyletic. So this is one of the problem with phylogenetic species concepts. Now that we are done with that, let's move on to taxonomy. So taxonomy is the science of classifying living things. So it is a quest to identify and relationships among organisms. More than 2000 years ago, the Greek philosopher Aristotle formally categorized all living things as either plants or animals. This eventually became genera. Then after that, Linnaeus started to use binomial classification for organisms. No two organisms can have the same scientific name, and they should the scientific name that they have should be same in all around the world. This actually will help to prevent confusion. So in a scientific name, the first word of a binomial name is the genus which the organism belongs, and the second word is called a specific epithet. So named species are organized into larger groups based on their characteristics, and it is evolutionary identical to categorized organism based on their shared derived characteristics, not ancestral characteristics. So organisms therefore group into 8 levels. Genera with similar characteristics are grouped into families, similar families into orders, orders within common properties into classes, and classes into phyla, and phyla into kingdoms. We have Archaea, bacteria, protista, fungi, plantae, and animalia as our kingdoms. In addition to these 8 levels of classification, we also have another classification called domains. So each of these hierarchical levels are called taxons, and we also have domains above these kingdoms. There are 3 domains which we'll talk about it in a little bit. So now we are going to look at an example of honeybee classification here. Each of the taxon groups organism by a set of characteristics. So here for European honeybee, the species level is called Apis mellifera, meaning the honey bearing bee. The genus level, it is actually a genus of bees which is called Apis. In a family level, we have Apidae. It's a bee family. All members of the family Apidae are bees. Some solitary, some living in colonies, such as Apis mellifera is. And then we have similar families gathered into order level. The order level is called Hymenoptera. Hymenoptera is a grouping that includes bees, wasps, ants, and sawflies, all of which have wings with membranes. And similar orders are grouped into class level. The class level here is Insecta, which is a very large class that are comprised of animals with 3 major body segments, 3 pairs of legs attached to the middle segment, and wings. And several of these classes are classified into phylum level. The phylum here is Arthropoda, and Arthropoda include animals. They have hard exoskeleton made of chitin and joint appendages. And several of these similar phylums are gathered into kingdom level. So kingdom here is Animalia, and the animals are multicellular heterotrophs with cells that lack cell walls. So we have this classification system for honeybee classification as an example. So new phylogenetics based on the molecular data reveal that linear hierarchy does not always reflect evolutionary history. Therefore, there are certain limitations of this hierarchy. Many higher taxonomic ranks, such as Reptilia, are not monophyletic. Therefore, they do not represent natural groups. A natural group is resulted from a group of organisms descended from a common ancestor. Any of the type of groups are artificial groups created by taxonomy. Any other class of groups are artificial groups. So when you look at Reptilia, it is not a monophyletic group, and many of the higher level of hierarchy are not monophyletic groups in Linnaean classification. So in Linnaean classification also, taxonomic groups may not represent clades that originated at the same time. One family may have diverged 70 million years ago. The other family might have had lot time to diverge and develop more adaptation. So the timeline of different clades are in taxonomic group may not represent the single each of these taxons may have diverged different timelines. Result of that, all of these families demonstrate different degrees of biological diversities and may limit the usefulness of making evolutionary predictions using these Linnaean hierarchies. So earlier I told you that there are beyond these 8 levels of hierarchical classification, we also have domains. So largest taxa are domains, and they are based on molecular data, and there are 3 main domains that we see today. So these domains include bacteria, Archaea, and Eukarya, which evolved from a most recent common ancestor called Luca, which is a last universal common ancestor that gave rise to these 3 domains. So these 3 domains are probably monophyletic, and bacteria are the most abundant organism on Earth, highly diverse. There are at least 12 to 15 major groups of bacteria based on ribosomal rRNA genes, and they played critical roles throughout the biosphere. They actually do carbon and sulfur cycling, extract nitrogen from air, they do photosynthesis. So bacteria is one of the most diverse groups in these domains. The Archaea is typically live in extreme environmental conditions. So a lot of them are called extremophiles, and they seem to be diverged from bacteria very early and more closely related to Eukarya. Evidently, because their ribosomal rRNA genes comparisons actually tell us that they are not close related to bacteria, but very close related to Eukarya. And most of the time, this microorganism has the ability to share genetic information via horizontal gene transfer, or HGT, with other microorganisms, which also might have helped to evolve into evolutionary diverse groups. So when you look at Archaea, even though there are very diverse group, all Archaea share certain key characteristics. Some of these include in this table. So one of the key importance characteristics are Archaea cell wall lack peptidoglycan. Peptidoglycan is one of the key things present, key molecule present in bacteria. So both Eukarya and Archaea lack peptidoglycan in cell walls. And archaeal lipids in the cell membrane have a different structure. They also have distinct ribosomal RNA sequences, and some genes process introns similar to Eukarya. And these Archaea also group into three general categories, such as methanogens. They obtain energy by using hydrogen gas to reduce carbon dioxide to methane gas. We have extremophiles that can grow in extreme conditions, such as high salt, high pH levels, low oxygen levels. And we also have non-extreme Archaea, which found in normal terrestrial, marine, and aquatic environments. Last domain is Eukarya. And when you think about eukaryotes, they appeared in fossil records much later compared to prokaryotes. Only about 2.5 billion years ago, they appeared in fossil records, and eukaryotic structure and function enable them to their cell to be larger and multicellular, and the root of the eukaryotic tree is still unconclusive. Nonetheless, Eukarya are grouped into 4 kingdoms, and you can see in this phylogenetic tree, these Eukarya actually separated into grouped into 4 kingdoms of Protista, Plantae, Fungi, and Animalia. So when you think about eukaryotes and how they were actually very successful evolving into multicellular larger organisms, the hallmark of eukaryotes is to be able to have complex cellular organization. This is highlighted by extensive endomembrane system. Both mitochondria and chloroplast are actually entered into the early eukaryotic cells by endosymbiosis. There is a serial endosymbiosis hypothesis which explains that one of the ancestral eukaryotic cell have engulfed free living bacteria that was having these mitochondria-like features, and then they started living in endosymbiosis. So mitochondria are the descendants of relatives of purple sulfur bacteria and the parasite Rickettsia, and also another eukaryotic ancestral cell has engulfed a chloroplast or a cyanobacteria, which develop into a chloroplast using endosymbiosis. And this is how the origin of eukaryotes is explained, as a series of endosymbiotic events happen in past. And also, when considering characteristic of eukaryotes, even though eukaryotic organisms are highly diverse, they share 3 characteristics that distinguish them from prokaryotes. One is compartmentalization. Compartmentalization actually have discrete compartments actually provide evolutionary opportunities for increased speciation within a cell. Bacteria and Archaea are distinct from eukaryotes because they lack compartmentalization. And like I mentioned earlier, eukaryotes develop extensive endomembrane system, and this endomembrane system actually evolved A nuclear membrane that is not found in bacteria and archaea accounts for increased complexity in eukaryotes. Physical separation of transcription and translation adds additional levels of gene expression for eukaryotes. We all know that in bacteria and archaea they don't have this separation of compartmentalization as nucleus. So their transcription and translation happens in the cytoplasm together. So it's one of the difference from eukaryotes to prokaryotes. And we also, eukaryotes have Golgi apparatus and endoplasmic reticulum that facilitates the intracellular transportation. This is also allow eukaryotes to be diverse and develop into many multicellular organisms, utilizing their molecular reactions to be separated from each other, which saves energy for them. And multicellularity actually lead to cell specialization and division of labor. And unicellular body plan tremendously successful. So unicellular prokaryotes and eukaryotes constituting about half of the biomass on Earth. Single cell has limit with the cell specialization. As we can see, when you have one single cell, it cannot grow larger because of the surface to volume ratio, and this is limiting the abilities of utilizing different niches on Earth. So being able to become multicellular allowed this organism to deal with environments in novel ways through differentiation. They were able to actually develop different metabolism mechanism to utilize different niches on Earth. Lastly, another shared characteristic of eukaryotes is sexual reproduction. And eukaryotic species as a group carry out sexual reproduction. Some interchange of genetic material occurs in bacteria. It's not regular and predictable. This happens through conjugation with sex pili and horizontal gene transfer sometimes, but it's not regular and predictable in prokaryotes. But in eukaryotes, reproduction occur, and then it is actually allow genetic material to be exchanged. Reproduction occurs only occasionally in many unicellular phyla. First eukaryotes were probably haploid, diploid rows in separate occasions. But sexual reproduction is also another characteristic of eukaryotes. So now we came to the end of this chapter. I hope you enjoyed this chapter. Please read through the chapter from the book and understand the material. And if you have any questions, let me know. Thank you very much.